Welcome to Manifested Publishers.
Hello learners, my name is Stephen Kariungi. I welcome you to our today's biology lesson, and we are still looking at the topic of transport in plants and animals, and we are doing the last part, and this is to carry out experiments to investigate transpiration. So, to investigate transpiration. So there are two experiments that we can carry out to investigate transpiration. Now, the first experiment involves getting a young shoot that is still growing, and then you get some cobalt chloride papers. The cobalt chloride papers are blue in color. And then you mount on both the upper side of the leaf and the lower side of the leaf. And then you leave them there for some time. After a few hours, or let's say after 1 hour, when you come to check the cobalt 2 or the cobalt 2 chloride papers, you will find that they have changed color from blue to pink. And they change from blue to pink when they come into contact with moisture. And therefore, basically, we can say that that moisture is coming from the plant or is coming from the leaf. So that means, or that is evident that the leaf is carrying out transpiration. So we'll have a sketch of a diagram. So to investigate transpiration, A, using cobalt 2 chloride paper. And what you are going to do here, we'll have a plant that is growing, or even a potted plant, a plant that is growing in a pot or in a container. We should not pluck out the leaves. The leaves should be left intact. And then you come and mount a blue cobalt chloride paper. So for one leaf, you can do it on the upper side, and the other leaf, you can do it on the lower side, on the lower surface, that is. So you repeat the same. So whatever is in blue is the blue cobalt 2 chloride paper. So we'll say that this is a blue cobalt 2 chloride paper. And of course, this is the leaf. That is the shoot. It is still growing. So the observations is that after 1 hour, the blue cobalt 2 chloride paper turns pink. That is the observation. explanation. This is because the leaf loses water that turns the blue anhydrous cobalt 2 chloride to pink. So when the cobalt 2 chloride is anhydrous, without water it is blue. But once you mount it on the leaf and then the leaf loses water, the water makes that cobalt 2 chloride to turn pink. So that's an indication that the leaf has actually lost water through transpiration. Another experiment also to investigate transpiration is using a potometer. Using a potometer, and the potometer looks like this. So this is a leafy shoot, a rubber bang. This is water. This is a water reservoir. A tap, a ruler. Then this is an air bubble. And this is a capillary tube. So that setup shows a potometer. And the setup is as it is labeled there. We get a young leafy shoot that is freshly cut underwater, and then mounted into this potometer that contains water, and then a rubber bang. The rubber bang makes sure that there is no evaporation. Then there is a reservoir to refill the water into the potometer with a tap. And then at the other end, there is a capillary tubing, a very thin tube that is dipped into water. So what happens? At the beginning, we introduce an air bubble. We introduce an air bubble, and then we set up the experiment as shown, and then it is left somewhere outside. And in this case, what will happen is the leafy shoot will lose water by transpiration. So in the process, as it loses more water from transpiration or through transpiration, it will be absorbing water from the potometer. And as it absorbs water and loses that water, the air bubble is sucked. And therefore, it moves over a certain distance along the capillary tube. So the work of the ruler is to measure the distance that is moved by the air bubble. And the distance moved by the air bubble can tell us how much water has been lost through transpiration. So we can say that observation, let's say after 2 hours, the air bubble moves a certain distance to indicate the amount of water lost by the leafy shoot. The explanation is that the rubber band prevents evaporation of water from the potometer. So we want to make sure that the water is only lost through the leaves or through the leafy shoot. The reservoir is for replacing water lost. It's for replacing the water in the potometer. The leafy shoot should be cut underwater to prevent blockage of the xylem tissue. By air. So basically that setup shows how we can investigate water loss by the leafy shoot using a potometer. So this device is called a potometer, and it's done by measuring the distance moved by the air bubble to indicate how much water has been lost. So the more the water loss, the longer the distance covered by the air bubble. We have also said that the rubber band makes sure that no water evaporates from the potometer. The reservoir is for replacing the water into the potometer. The ruler, of course, for measuring the distance. The leafy shoot should be cut underwater to prevent air from blocking the xylem tissue. So the assignment So the questions in the assignment, number 1, name the device that is used to measure transpiration. Number 2, what happens to blue cobalt 2 chloride when it comes into contact with moisture? And number 3, why should the leafy shoot, why should the leafy shoot, this is a shoot, why should the leafy shoot be cut underwater before putting it in the potometer? So we are going to stop there. Until next time, goodbye.